Hi everyone, I'm going to be going over some of the answers to this test review. So starting with drawing a segment of DNA, I'm going to do it sideways because it fits better that way. I usually start by drawing the pentagons, I guess, because that's the hardest part for me to draw. Yeah, okay, we did that all right. And those, of course, are the sugars or the deoxyribose sugars. Then we can put in our phosphate groups and connect them. Oh, I'd also like to point out that notice I did draw the pentagons going in opposite directions because of that whole anti-parallel thing. Then our bases are in the middle, so maybe let's make this A and this T and this C, and these aren't quite lined up right, but that G. And then for labeling, again, this was the phosphate, and each of these in the middle is one of the bases, and the two together would be a base pair. Okay, to circle one nucleotide, I'm going to need one phosphate, one deoxyribose, and one of the bases. So make sure you don't circle both bases. It's just one of each thing makes one nucleotide. Okay, DNA replication is semi-conservative. That means that when DNA replicates, if blue is my original DNA, when it gets copied, the new DNA molecules each contain one strand of the original DNA and one strand of newly created DNA. Right, these are all kind of definitions, so I just encourage you to review that DNA replication section again so that you know what all of these things do. Okay. And then all of the chromo stuff. So, chromosome, duplicated chromosome, so also a chromosome, centromere, homologous chromosomes, homologous chromosomes that are duplicated, and then each one of these, this one and that one, for example, could be sister chromatids. So each half of the duplicated chromosome is one chromatid. And then chromatin is the substance that the chromosomes are made of, which is a combination of DNA and protein. The DNA kind of wrapped around the proteins, like those histone proteins, in order to keep it sort of organized and not tangled. A 2N cell, a cell with two complete sets of chromosomes, is called diploid, which is what most of your cells are at any given time. A 1N cell with one set of chromosomes is called haploid. So somatic cells, so all of the regular cells, are diploid. Gametes, the eggs and the sperm, are haploid. And that makes sense because if you put one egg and one sperm together, you get like the full set of DNA, which is what the baby would have. End results of mitosis: two cells, same as the original cell. How many chromosomes? You could say 2N, a full set, diploid. Meiosis, how many cells in the end? 4. Different from the original 1N or haploid, so half the chromosomes. Why are the cells resulting from meiosis different? Two things. One part is the crossing over that occurs with homologous chromosomes exchanging bits of genetic material with each other. Let's see, I have to draw this kind of like that. which sort of mixes up the genes. And the other thing is that when they first divide during the first anaphase, so these will go that way and these will go that way, it's random which of the chromosomes ends up on which side of the cell. So we call this independent assortment, where which genes end up with which other genes is random, how they line up. And the fact that these line up in pairs, so they split the genetic information in half on that first division, that's what causes our meiosis cells to all be different from each other. Okay, cell cycle, circle thingy, draw a circle, and then an offshoot for G0. You can do G1, S, G2, and M. And I didn't draw these perfectly, but G1 should be the largest and M should be the smallest. The checkpoints are located at the end of G1, at the end of G2, and in the middle of M. What happens at the different phases of the cell cycle, I think you can review that from the video. It's important to know that S is when the DNA replication occurs. So that's when the chromosome number in the cell doubles temporarily until the division occurs. Cancer cells and normal cells, lots of differences there. The main thing is that cancer cells just divide out of control. So they divide. They ignore the checkpoints. They ignore the rules about how they shouldn't divide if there's not enough room for them to divide, the density-dependent inhibition, that they shouldn't divide if they're not attached to things, which allows them to metastasize. They generally have a lot of mutations, and because they're dividing so quickly and sort of recklessly, those mutations will continue to build up, and they'll build up more and more as those cells divide. Cyclins and cyclin-dependent kinases, so those are chemical molecules inside the cell that regulate the passage of the cell through the cycle. Cyclin-dependent kinases are enzymes, and cyclins are molecules that have to be present to activate those enzymes, so like cofactors. The cyclins concentrations rise and fall throughout the cell cycle, and different cyclins are higher at different times. And when the appropriate cyclin is high enough, it will activate its kinase, which will then proceed with whatever its job is in the cell cycle and moving the cell through that cycle. They're especially important in regulating the G2 checkpoint. The role of growth factors in regulating the cell cycle, those are especially important in regulating the G1 checkpoint, and growth factors need to be present outside the cell in order to stimulate it to divide. The M checkpoint, the only regulation thing on that is that all the chromosomes are lined up in the middle, and they're all attached to their spindle fibers so that they can be pulled apart correctly when the cell moves into anaphase. Right, and then do review the meiosis and mitosis videos. Make sure that you can recognize pictures of the cell in different phases of the cell cycle and you know which one's which, and that you know the difference between how they divide in meiosis versus how they divide in mitosis. Right, as always, if you have any questions, please make sure to ask me.